My name is Jason Mikio-Hunter, and I'm going to talk about our work implementing different spontaneous rate classes of auditory nerve fibers in the CARFAC model as part of the Australian Future Hearing Initiative. The CARFAC model is a digital model of the human inner ear. It uses a cascaded filter bank to represent the basilar membrane motion and cochlear filtering, here shown at the top as boxes with H's. This basilar membrane motion is then actually used as input to the inner hair cell stage in each of the frequency channels, where the inner hair cell stage output is the actual final output of the cochlea, known as the neural activity pattern or NAP, but also provides input to automatic gain control that directly feeds to the outer hair cell stage that is coupled to the filter bank. The main difference between CARFAC V2 and V3 can be found in the inner hair cell stage. In CARFAC V2, the transduction current would pass through a first capacitor stage that represented essentially membrane filtering of the inner hair cell, leading to a receptor potential. This would then be used as an input to a second capacitor stage that acted as a synaptic gain control. The final output being passed through a low-pass filter and generating a NAP, which could be considered as equivalent to mean synaptic drive or mean firing rate for a particular frequency channel. We introduce three different fiber types. This is achieved by taking the receptor potential from the CARFAC V2 and passing it to three parallel pathways that represent the different auditory nerve fiber types. They differ mainly at their onset in the sigmoidal transfer functions that are shifted along the receptor potential axis here, and these then transform the signal into a synaptic drive that will lead to different release probabilities that, combined with the low-pass filter and the number of fibers in each different class, leads to a number of spikes that can be here converted into a mean spike rate. As for above, this is for a 40 dB 4 kHz tone, showing how the high spont rate fibers at the top actually responds more prominently than the low spont rate fibers at the bottom. While the inner hair cell stage has changed drastically, the resulting data still compares well to both other models and electrophysiological data. Here we can see there's some response to a 4 kHz tone at 70 dB across all fiber types for the 4 kHz frequency channel in the CARFAC V3, and can see how the actual time course and adaptation is very similar to that seen in both the phenomenological models of Jeleni and Bruce and the biophysical models of Verhulst. And we then compare responses to 4 kHz tone at different intensities. We can see that the different responses of the high, medium, and low spontaneous rate fibers in blue, orange, and green respectively, with the mean rate shown in black, which is equivalent to the output of CARFAC V2. As you can see, both the actual numbers are quite similar at both the onset and the steady state, where the steady state is the mean rate for last 40 milliseconds. But also you can see saturation at the highest stimulus levels, which is both characteristics that we would want to reproduce in CARFAC V3. But now that we have a CARFAC V3 model with initial parameters that are we're relatively happy with and that are suitable for testing, how do we go about tuning those parameters, especially with the purpose of generating audiometric thresholds for test tones inquired? Well, the first step is to actually to apply a middle ear filter to CARFAC V3. In the previous case for CARFAC V2, we used the D prime metric, a separability index, which compared the number of spikes in response to test tones compared to the number of spikes in response to silence. For that to happen, we needed to transpose the NAP output of CARFAC V2 into essentially a high spontaneous rate fiber. This led to compensations at the different audiometric frequencies as shown in the purple line. However, this was only using a D prime in a single channel, and now that we are aiming with CARFAC V3 to incorporate different fiber types, but also to be more inclusive of all the information from across the cochlea, we now use a sum D prime, which takes the D primes from each channel, each frequency channel, and each fiber type, and then sums them. This leads to compensation values that are much flatter than had previously seen for CARFAC v2, but also much flatter than seen for pre-existing middle ear filters as used in previous models. Result, we see that when we actually apply a pre-existing middle ear filter from Verhulst 2018 with a gain of 9 dB, allowing for a neurometric of 1 at the lowest and the highest audiometric frequencies, we actually find an increased sensitivity around 3 kHz. This increased sensitivity does not seem to be only associated with the interaction of the middle ear filter, its gain, and the CARFAC V3 sensitivity, but also seems to be associated to the D prime metric itself and the way we calculate it. So the D prime metric actually compares the difference in the means between firing and spontaneous rate, here shown in orange, versus the actual mean rate of the of firing, which is shown in blue here to the test tone. And at 1/10 of 25 Hz, you can see the oscillatory nature of it means that despite actually having much larger deviations in firing rate, 3 kHz, that it actually leads to a much smaller D prime neurometric compared to 3 kHz, which has this asymmetric response due to the low pass filtering of the inner hair cells, leading to a greater DC component. As a result of this, we have moved to using the structural similarity index measure that's typically being used to compare images and their respective quality. This is a weighted product of 3 components: the luminance, contrast, and structure, leading to the total similarity structural index measure. It has been adapted for neural signals and has been used within the auditory field by Ian Bruce's group. Rather than using the SSIM with cochleograms, however, we're using them with stabilized auditory images. We compare the stabilized auditory images for test tones versus silence. Here you can see these SSIM matrices, which are the product of the luminance, contrast, and structure, for 125 Hz tone versus silence at 4 different levels: 30, 40, 45, and 50. At 30, you can see that clearly there's no structure at all, and indeed it is below the threshold, and exactly the same. However, if you look at 40 dB already, you can start to see the structure in the SAI that is associated with that oscillatory firing rate at 125 Hz. So we believe that using the SSIM and the correct scaling factors, we will be able to improve the sensitivity at these 125 Hz, at these low frequencies, and matching those at 3 kHz. More other the SSIM neurometric will allow us to more effectively move from these audiometric thresholds and test tones in quiet to tone in noise and other super threshold stimuli by using the 3 different components and changing their scaling or weighting and scaling coefficients. And once we are settled on parameters for the SSIM that allow us to maintain a good sensitivity across the different frequencies, we can move on to actually changing the necessary inner hair cell parameters that might be required to actually effectively get audiometric thresholds of a normal hearing individual. I thank all my co-authors on this study: Dick, who's been involved in every single part of the study, Rob and Hong Lin, who have been vital to understanding how to use the CARFAC and to run it in JAX, as well as individuals who are not named but have been key to actually helping with the stabilized auditory image and structural similarity index measure. Zhanghai Wu, who's been looking at the parameters that are going to be necessary to achieve audiometric thresholds, as well as Mariam Hassini, who's been using it as part of her DNN in the cochlear implant world. I'd also like to thank Ian Bruce, who provided Mariam with the code for the SSIM. Additionally, I'd like to thank all partners of the Australia Future Hearing Initiative that have come together to make this project possible. Thank you.